Thank you everyone for having me here today. It's really an honor to be here and talk about my work with you guys. So I'll start with just a short introduction to obesity in general. Currently in the US, obesity in itself is a major problem. More than 1/3 of adults in the US are currently obese. And this is really a multifactorial disease that can be caused by and contributed to by many different factors. So this includes energy intake, energy expenditure, psychological factors, the microbiome, environment, culture, socioeconomic status, and genetics. So in Rudy Leibel's lab, myself and others are really interested in better understanding how genes contribute to the regulation of body weight. And specifically, I've been working on trying to understand how the genes that are lost in Prader-Willi syndrome contribute to the regulation of body weight. So it's important to understand that the genes that control body weight are mostly acting in this very important center of the brain called the hypothalamus. And this is, in human adults, about the size of an almond. It's a very small part of the brain, but it's very important for homeostatic processes in general. So this includes body weight regulation, sex drive regulation, thirst, temperature, and sleep. And we know from some really nice studies that were carried out a long time ago, some dating back to the 1940s, that if we disrupt the hypothalamus, specifically the regions of the hypothalamus that are important for anorexigenic processes, that you can have a mouse or a rat that previously was perfectly healthy, normal body weight, and after disruption of the hypothalamic centers, you end up with a mouse or a rat that wakes up from surgery ravenous, always looking for food, and as a result becomes very obese. So this really illustrates the importance of genes and their products acting in the hypothalamus in the regulation of body weight. Now, to kind of zoom in on the hypothalamus here, there are discrete nuclei within the hypothalamus that contain genes and proteins that contribute to body weight regulation distinctly. So our fat is where we produce the adipokine hormone called leptin. And this hormone is produced almost in exact proportion to the amount of fat mass that one has. So leptin is secreted from our fat cells. It travels through the blood and then can act on its receptors in the hypothalamus. And cells expressing leptin receptor make other products, including CUMC, NHLH2, and prohormone convertase 1. And from these cells, you end up with an anorexigenic product called alpha-MSH, which can then travel to these second-order neurons, also in the hypothalamus, and produce BDNF. Now, this is important because we know that if you disrupt this pathway with, for instance, loss of function mutations, you end up with obesity in humans and in rodent models. And this includes some of these monogenic and syndromic obesities that I've listed on this slide here. So just to further highlight the importance of this pathway in body weight regulation, you can see that messing with these genes in this pathway results in hyperphagic obesity in humans and in rodents. So one of our major questions is if the genes in PWS could be acting in this pathway. I want to give just a brief introduction to some of the genetics involved in Prader-Willi syndrome. So this is a pretty complicated region of the genome, as Teresa will explain to you in more detail later. You can see here that genes shown in blue are expressed only from the paternal allele, while genes shown in pink are expressed only from the maternal allele, and then the genes shown in green are expressed from both alleles. So most instances of PWS are caused by these type 1 or type 2 large deletions on chromosome 15q. However, recently, researchers have identified a handful of patients that have much smaller than typical deletions, but still present with all of the major phenotypes of Prader-Willi syndrome. And if you map the overlap among these deletion regions, you can find a minimum critical deletion region that's enlarged here that's sufficient to cause all of the major phenotypes of PWS in humans. And this region includes 3 genes, all of which are non-coding, which means that they don't make proteins, they make RNA. And these genes are SNORD109A, SNORD116, and IPW. So we were fortunate enough to be able to obtain a skin biopsy from the patient with the smallest microdeletion identified to date. And I will explain more how we utilized this skin biopsy to help us understand the molecular pathogenesis of PWS. But first I want to go back to SNORD116. So we hypothesized that this gene could be a candidate gene by loss for Prader-Willi syndrome. SNORD116 is a small nucleolar RNA, so this means that it's thought to be expressed in the nucleolus. And the nucleolus is simply a subcompartment of the nucleus, which is the part of the cell that houses your DNA. Most of these SNO RNAs are acting in the modification of ribosomal RNAs. And the ribosomal RNAs, or ribosomes, are basically cellular machinery that make proteins. However, basically you can forget about that because there's no known ribosomal RNA targets for SNORD116. So we really don't know at a cellular level or molecular level exactly what it's doing. We do know that it's highly expressed in the hypothalamic, arcuate, and paraventricular nuclei, which if you remember the slide with all those red boxes, that is where that is. So this is very telling that it has a high expression pattern in an area that we know to be important for the regulation of body weight. We know in humans, as I mentioned, that the loss of SNORD116, IPW, and SNORD109A are sufficient to cause all of the major phenotypes of PWS in humans. And furthermore, this is the mouse model that Teresa was alluding to. Mice that have a paternal deletion of just SNORD116 do display many of the neuroendocrine features of Prader-Willi syndrome. This includes low growth hormone and growth deficiencies, impaired motor learning, hypoinsulinemia, high levels of circulating ghrelin, and hyperphagia per lean body mass. However, these mice, and to date, all published existing models of PWS mice do not demonstrate the obesity that is so typical of Prader-Willi syndrome. So we wanted to try a different approach and to use stem cell technology to create an in vitro or an in the dish model of PWS. So the way that we do this is by obtaining skin biopsies from patients, and we then take those skin biopsies into the lab and we grow the cells in a dish. Then we can add what are called reprogramming factors to those skin cells, and that will cause them to reprogram to what are called induced pluripotent stem cells. These induced pluripotent stem cells, or iPSC, can, in theory, differentiate to any cell type in the body. To study PWS, we're most interested in examining neurons. generated from induced pluripotent stem cells. So I won't show you any of the data for this, but just to summarize and tell you that we made these IPS and IPS-derived neurons, and we characterized that they do indeed retain the molecular signature of PWS, and therefore are likely a good model to study the molecular mechanism of PWS. So from our IPS-derived neurons, from individuals with PWS and unaffected control individuals, we performed what's called RNA sequencing. So this means you can analyze all of the genes that are being expressed in a cell at a given time all at once, which is very cool. So from this RNA sequencing study, we actually found that 2 genes that were in this top hits of downregulated genes were NHLH2 and PCSK1. So I'll explain more what these genes are. PCSK1 is the gene name, and we call the protein PC1. I didn't clone this gene, so don't blame me for the confusing nomenclature. Not my fault. But this is called prohormone convertase 1. And some people call it PC1/3, which is even more confusing, so we'll just stick to PC1. Anyway, as its name suggests, this is a prohormone convertase. And that means that this gene, which makes a protein that is an enzyme, is very important for the maturation of hormones from their generally inactive, immature form to their bioactive, mature form. And human patients and mice that have mutations in PCSK1 have hyperphagic obesity, they have hypogonadism, low growth hormone and short stature, and impaired prohormone processing. I also mentioned that we found that NHLH2 was downregulated as well. This is another gene that is what's called a transcription factor. And the NHLH2 transcription factor is actually very important for increasing cellular levels of PC1. If you delete the NHLH2 gene in mice, these mice have hyperphagic obesity, they have hypogonadism, they have growth deficiencies in their early adolescence, and they also have impaired prohormone processing. So this set of symptoms should sound very familiar to people in this room. The question now is there, where's my pointer, impaired prohormone processing in PWS? So before we move on to look at more impaired prohormone processing, we did go back and confirm these results in an independent differentiation, and we do see that NHLH2 is downregulated at the transcript and protein level in our PWS neurons as compared to unaffected control, as well as transcript levels of PCSK1 and PC1 protein. So, interestingly, hormones that are processed by PC1 are dysregulated in PWS. So this includes ghrelin, the gonadotropin, pro-growth hormone releasing hormone, which is important in the production of growth hormone, and proinsulin. And this suggests that the deficiency or the observed deficiency in PC1 could be very important for the major endocrine phenotypes in Prader-Willi syndrome. So we hypothesize that in the setting of the paternal loss of SNORD116, this causes a downregulation of NHLH2 and PC1, and in the setting of a cellular deficiency of PC1, prohormone processing will be impaired. So in the case of prohormone candidates for body weight regulation. We actually have a few right now. Impaired processing of really any of these prohormones could lead to hyperphagia, increased body weight, and obesity. PWS patients are known to have high circulating levels of ghrelin. This has been replicated in many clinical studies. And interestingly, the antibodies used, so antibodies are just the technology that you use to measure hormones in the blood or in cells. The specific antibody that's often been used to measure ghrelin levels in the blood of PWS patients can actually identify both progrelin and ghrelin, and we've done this with those antibodies used in those studies in our lab and found this to be the case. So if this is the case, this means that we could have a situation where we're detecting increased total ghrelin, which could really be reflective of possibly an increased fraction of progrelin. And we know that mice that don't have PC1 have increased progrelin and decreased mature ghrelin. Furthermore, if you have impaired processing of progonadotropin-releasing hormone, you will have hypogonadism. Impaired processing of pro-growth hormone-releasing hormone can lead to decreased growth hormone and short stature, and impaired processing of proinsulin can lead to hypoinsulinemia and hyperproinsulinemia and later development of type 2 diabetes. So we had the question, we saw the PC1 deficiency in our cellular or in the dish model of PWS, but is there impaired prohormone processing in an in vivo or a whole animal model of PWS? So we turned to the SNORD116 deletion mice to try and answer this question. We injected these mice with glucose, and 30 minutes after the injection of glucose, so injecting glucose into you or me or into a mouse causes insulin levels to increase, right? And what we found is that in our SNORD116 deletion mice, the ratio of proinsulin to insulin was really majorly elevated. And this suggests that proinsulin was not being processed properly in these animals. We also then isolated their islets into a dish to take them out of the context of the whole body model, and also found that the ratio of proinsulin to insulin was dysregulated. Furthermore, we were also able to obtain plasma samples from people with PWS, one patient with a PCSK1 mutation, and age and BMI matched unaffected controls. And we found that there's at least a trend towards an increase in the proinsulin to insulin ratio, suggesting that proinsulin processing is impaired in people with Prader-Willi syndrome. From these isolated islets from the mouse, we also see that PCSK1 transcript and protein are downregulated, as well as its sort of companion processor, PCSK2 transcript and protein. We next looked at the progrelin axis in these mice. And so as I mentioned, PWS patients have increased circulating ghrelin levels, and these mice also have increased circulating ghrelin levels. So this is total ghrelin. If you look at their stomachs, the SNORD116 deletion mice don't express SNORD116 since it's deleted, but you see nice expression in our wild type or control animals. We see a downregulation of PCSK1 transcript and protein in our SNORD116 deletion mice, but we see an increase in the gene expression levels of pre-progrelin. So this is encoding the full-length progrelin protein. And when we look at processing of progrelin at the protein level in the stomachs of these mice, we see an increased ratio of progrelin to mature ghrelin in the stomachs of the SNORD116 deletion mice. And you can see that this effect is intermediate to that of the PC1 null mice. And this could be expected since we don't have a situation of complete PC1 ablation, rather a downregulation of PC1. So there still is some PC1 around, just not in the same levels as our wild type or control mice. Next, we looked at the pro-growth hormone releasing hormone axis in these mice. And just as a sort of intro to this axis, so pro-growth hormone releasing hormone is made in my favorite part of the brain, the hypothalamus, and it's processed to growth hormone releasing hormone by PC1. So this growth hormone releasing hormone that's made in the hypothalamus then travels through what's called the hypophyseal portal system to the pituitary, the anterior pituitary, where it can then act on the cells that make growth hormone. In the setting where you have sufficient quantities of growth hormone releasing hormone acting on the growth hormone producing cells in the anterior pituitary, you get growth hormone released into the blood. And then this growth hormone in the blood can act on its receptor in the liver and then stimulate production of IGF-1, which is important for many processes, including growth. Our SNORD116 deletion mice have lower body weights at weaning and throughout adulthood, and they also have decreased body length at 1 month of age and 6 months of age, which for a mouse, 6 months of age is quite old. These mice have decreased circulating IGF-1, so that's the sort of lowest point on that slide I was showing you before, indicating that something in the growth hormone axis is probably gone awry. Their liver transcript levels of IGF-1 are also decreased. However, when we looked in the pituitary at growth hormone levels, we saw no difference. So this suggests that the pituitary is able to make growth hormone just fine, but maybe it's not getting the signal to release growth hormone into the blood, as is reflected in this IGF-1 level. So we then took out the hypothalamus of these mice, and we looked at the ratio of pro-growth hormone releasing hormone to its mature product, growth hormone releasing hormone. And we found that this ratio was elevated, at least a trend towards being elevated, in the SNORD116 deletion mice, suggesting that processing is impaired in the hypothalami of these mice. So this suggests that PC1 deficiency, as I mentioned earlier, could really be driving some of the major endocrine phenotypes of Prader-Willi syndrome. So using our in the dish and in vivo mouse models, we found that in the setting of paternal deficiency of SNORD116, we have a downregulation of NHLH2 and PCSK1, and we were able to demonstrate that progrelin processing is impaired, pro-growth hormone releasing hormone processing is impaired, and proinsulin processing is impaired. And we also were able to look at proinsulin processing in plasma from humans as well. The axes that are shown in the dotted lines still need to be thoroughly investigated. However, we do hypothesize that the PC1 deficiency is really what's driving the hyperphagia and obesity. So in conclusion, I'm going to give the sort of more technical conclusion slide first, and then I'll give you a more maybe, toned down conclusion slide, but for any of the, more technical people, you get this one first. So we were able to reprogram PWS skin cells to induce pluripotent stem cells, and these retain the molecular signature of PWS, including the maternal imprinting on chromosome 15. And we found that when we differentiate these iPS cells to neurons, that PCSK1 and NHLH2 are downregulated in the PWS neurons as compared to unaffected controls. We also found that these transcripts are downregulated in the hypothalami of SNORD116 deletion mice. These mice have impaired prohormone processing of proinsulin, progrelin, pro-growth hormone releasing hormone, and this is associated with tissue-specific deficiencies of PC1 in each of these tissues. We also found that the ratio of proinsulin to insulin at least trended towards an increase in plasma from fasted individuals with PWS as compared to unaffected controls, suggesting impaired proinsulin processing in people with PWS. So these, in the aggregate, suggest that impaired prohormone processing could really be driving the major endocrine phenotypes of PWS, including hyperphagic obesity. So what does this mean? Essentially, what we did is we generated a cellular model of PWS in a dish, and then we used this to investigate the cellular mechanisms in PWS. This type of a model system can also be used for things like drug screening and drug testing. From our RNA sequencing experiments, we identified a candidate molecule, which is PC1, that is underactive in PWS. This identification of the downregulation of PC1 can potentially link the loss of these genes in PWS to the symptoms at the patient level. So this might be a link between the genetics of PWS and the hyperphagia. And importantly, PC1 is potentially a druggable target. So this is what we're most excited about now, and we really want to work on strategies to potentially increase cellular levels of PC1, and we hypothesize that if we can do so, this may mitigate the hyperphagia in PWS. So there's still a lot of questions that we still have, including can we identify compounds that increase PC1 levels, as this is a potential therapeutic strategy? We still want to further understand the real molecular mechanism of hyperphagia in PWS. So we think that PC1 is involved in the hyperphagia, but we're still not sure exactly which candidate prohormone in the hypothalamus is really responsible for this phenotype, and it could be a combination. So these are things we're still working to understand. Furthermore, we also want to understand the real mechanism by which the loss of paternal SNORD116 is really, how does it actually cause the downregulation of NHLH2 or PCSK1? In a wild type cell, does SNORD116 physically interact with these genes? These are still big questions that we would still like to answer. And furthermore, we also are very interested in trying to generate an obese model of SNORD116 deletion. If we have a mouse that actually is becoming obese, we can really much better test any potential drug candidates for increasing PC1 levels. So some of the ideas that we have to do this are knocking SNORD116 out in an adult mouse. So you sort of skip that phase of growth delay. And we also will consider knocking SNORD116 out in the specific neurons within the hypothalamus that I was talking about earlier. Okay, so thank you all for listening to me, and I'm happy to take questions. And I first just want to thank all of you guys for your continued support of this project. And it really would not have been possible to do this work without all of your support. I also want to thank my mentor, Rudy Leibel, who has been so wonderful to work with, and we really have a great lab environment at Columbia. And I want to thank all of so yeah, here's a picture of our lab. It's our fearless leader, Rudy, in the middle. And we have really a great lab environment and a really nice team to work with. I also want to thank our collaborators. I'm sure many of you are familiar with Dan Driscoll. He has been great to work with. I'm going to keep talking. And really helped us get started with this by sending us some of the skin biopsies from patients with Prader-Willi syndrome. I also want to thank Dr. Taubert and her colleagues at INSERM. She sent us the very important microdeletion fibroblasts that we were then able to work with. And also, Dr. Butler, who probably many of you know that name as well, was really great. We were like, we really want to get some plasma from people with Prader-Willi syndrome, and within the next week, we had like 16 samples, so that was great. That never happens. Also, Dr. Day and Dr. Levy at University of Sherbrooke and CUMC also assisted us. So thank you, and I'll take questions.
Are there any drugs that we know of that already could perhaps be used on PC1?
We think there may be, and we're in the very early stages of testing that.
So one of the slides, it looked like not only were the deleted, the SNORD116 deletion mice showing decreased length, but also decreased weight throughout life. Is that right?
Yeah.
How does that line up with the theory that the deleted SNORD116 and then the downregulated PC1 would relate to hyperphagia if the mice have lower weight?
Yeah, so this is a great question, and this drives me totally crazy every day, that they don't get fat. Why? So it's complicated, and mice are different from humans. Let's see. Well, let's stay on the slide. So PC1 null mice show essentially the exact same growth curve. They have decreased body weight throughout life and decreased body length. Mice that have a mutation in PC1 basically have less of an impaired growth hormone axis, and these mice don't have growth deficiencies, but they become hyperphagic and obese. So what we think from those data and then relating it to our SNORD116 mice is that the levels of growth hormone in the mouse early in life seem to be very critical. And if they're too low, it seems like the mice never are able to overcome that and really manifest the hyperphagic obese phenotype. And this is why we really want to delete SNORD116 later in life. So if you look at the black line, this is the wild type mice, and you can see that between 3 and 6 weeks of age, that's when they're really doing a lot of their growth. And then other than that, it's just sort of the middle-age weight gain, if you will. But we think if we can delete SNORD116 after this major growth spurt, we could maybe miss that critical window where growth hormone is really important and maybe get an obese hyperphagic mouse.
Are the deleted SNORD116 mice overfeeding? They're just not gaining weight?
Yeah, so if you fast them, so we remove the food overnight from the mice and then let them eat for a timed period, whatever they want, they eat the exact same amount of food as wild-type mice. But the wild-type mice are much larger than them. So if you imagine comparing, a 100-pound person to a 300-pound person, those people, based on their body size, don't need the same amount of food. So if the 100-pound person eats as much as the 300-pound person, assuming they're both eating to, repletion, the 100-pound person is overeating. So we see that hyperphagic phenotype, but it would really be better for studying things if we had a mouse that actually got obese. Mm-hmm.
Thank you.
Did you test the processing of nucleobindin 2?
No. No, I didn't. Mm-hmm.
Thank you, Lisa.
Thank you.